Hi, I'm Trace, and I'm really excited to present my research today. I'm innovating microbiological water quality testing. So I want to start with a question. Have you all heard you aren't supposed to go in the water like 3 days after it rains? Yeah, well, you're not if this is your first time hearing about it. That's because when it rains, it flushes a lot of contamination from the land into the ocean, and it is more likely to get you sick. I'm particularly focused on the microbiological side of that, the bacteria, the viruses, the pathogens. So not just for when it rains, but for routinely, we monitor our beaches for water quality. Is it safe to swim in the water? Our target here is a bacteria called Enterococcus species. That isn't the one that's particularly getting you sick, but it co-occurs with other viruses and pathogens. So Enterococcus is one of our commensal organisms. It is inside every single one of us right now. So like most bacteria, it's an opportunistic pathogen. All bacteria is just waiting to get to you, but it's fine. So how this water quality monitoring occurs, we physically collect that water from the ocean. It gets transported to a laboratory for analysis results. We can then use those results for our beach management decisions. Now, a high Enterococcus result isn't usually grounds for closing a beach, but it's good to like post a warning. They're like, hey, that beach is kind of gross. Consider if you want to swim in it. So also, this is Enterococcus right here. I took this picture under the microscope. It's about 1,000 times blown up. So small problem with this, 24 hours in the laboratory. So a little lag between sampling monitoring. So the county here, they sample on Mondays, usually get to the samples to the lab by Monday afternoon, results out by Tuesday. The report drops in my inbox on Wednesday. So with that, my question to you, is it safe to swim? You can find out tomorrow. So why is this process so long? What's going on in the lab for 24 hours that they need 24 hours to analyze these results? Well, that is because bacteria are very small. They are hard to count. They are hard to quantify. You can do it. It's expensive. It's hard to do. So we need to amplify them in some way to some kind of countable result. So there are 3 federally approved laboratory methods for doing this to grow the bacteria so we can count them. 2 to 5 days, 24 hours is our fastest. So what's going on there with our fastest one? We have this water sample here. We mix it with a really special food called a substrate. It's connected to a fluorescent molecule. Our target bacteria possesses an enzyme that's capable of breaking apart that molecule, releasing the fluorescence over time. To make a quantity of it, we have divided it into several compartments. Glows blue, it's got Enterococcus in it. Does not glow blue, no Enterococcus. We can then throw that into a formula to get the quantity of bacteria in the original sample and use that for water quality decisions. This is what it looks like in real life. I also took this picture. That's very pretty, very beautiful research. So what's going on in this build? I am not the only one trying to address this 24-hour issue. There's a lot going on. People are like, hey, why don't we analyze the organism DNA instead? Why don't we look for alternate indicators that aren't Enterococcus? What about direct pathogen monitoring, like getting what actually makes you sick? Turns out that's quite hard to do. And then our modelers out there are saying, why are we sampling it at all? Why don't we just use pool models to tell you your risk of like being in the water? And me, I have a professional laboratory background, so I'm like, hey, why don't we try to address this from the perspective of why don't we make this method faster? It'd be a good idea. So I wanted to do that. I knew exactly what I wanted to do, not how to do it. So one day, my advisor over here slides me a paper on microfluidics. First time I ever heard of it. But it's really simple. It's simple. Microfluidics is just manipulation of fluid at the micro scale. So small scale fluid. You've seen this before. These are your ancient printers, different like tests to them from pH tests, pregnancy tests, COVID rapid tests, your ballpoint pens, dry erase markers, anything that involves the small manipulation of fluids. Because when you put fluid that small, it has a bunch of really cool properties. Basically defies the force of gravity, capillary force takes over. People are using it for a wide range of biological, chemical, different kind of engineering applications. When you put it in the context of the laboratory, it does impart a lot of advantages to laboratory tests. So improved method performance, less material consumed, possibly cost effective, really versatile. But what I saw in these papers, faster. So dug into the literature a little more, 3 successful ones I found that have successfully both cultured and quantified bacteria using microfluidics. One for salmonella, for the case of like that, someone gets you sick when you foodborne illness, that's your salmonella. E. coli, which y'all know and love, another one of our commensals that's also inside of us right now. So 2 groups today with E. coli. So why is smaller faster? One of the E. coli groups said, for example, so time is going in this direction. They said, if you take 1 bacteria here and you put 3 foods with it, over time, let's say it releases, for example, 3 fluorescent molecules. They're saying take that same exact bacteria, put it down in the microscale with those same 3 foods, same amount of time, only 3 fluorescent molecules still, but they're saying because it is against such a smaller background, it is faster to detect. This group says smaller is faster. So I proceed. So easy peasy. All I need to do is build a microfluidic device capable of quantifying Enterococcus. And then I also need to build a sample pre-treatment method. The unfortunate part about microfluidics is not super great for environmental samples. They're really small channels, not good for like sediments, for sand. Our bacteria has a pretty low concentration, so I need to find a way to concentrate it. So easy, do this, do that, PhD, I'm done. So I'm going to talk to you today about designing this device. So we're going to start with just the designing part. It's an iterative process. I have a cool idea. I have now learned how to draft in computer-aided design software to produce that idea into a file, which I then can send to the 3D printer. We're using a really cool fabrication technique for microfluidics where we 3D print the part, cast it fully in silicone, and then dissolve out that 3D printed part. Then we can use that now created device to test like fluid. How does it handle fluid? Can it grow bacteria? Is it killing all the bacteria? And then we can take, this is image analysis, so we're taking pictures of this, take that image data, and then make it quantitative through image analysis. Now, something to note, not all devices that have cool ideas make it to the full phase. A lot of times I design it, I fabricate it, it breaks. I design it, I fabricate it, I test it, it was terrible. And so iterative, this has been a lot of work, but what I want to tell you about today is my current iteration. So I do have something. This is what it looks like in real life. So it's clear, it's squishy, it's made of silicone, it can pass, this is it with food coloring inside, passes fluid right through. Now, I want to tell you about some features of this device. So stay with me, keep this in your minds though. Okay, a couple features. One single channel, fluid goes in on this side, out on this side. We're putting bacteria, water, the bacteria food it needs inside there. I have folded it using heat into this three-dimensional structure before I cast it in silicone. This features a 4-stage silicone casting. I've got 2 squishy layers, squishy layer, squishy layer, with a firm inner core. I can control the compliance, the squishiness of the silicone with those ratios. Why is this? Why am I telling you about this? Why is this important? It's because that is what we are using to physically isolate. Remember, I told you you quantified it based on putting the water into compartments. So I need to make isolated compartments out of this device. So we are physically pinching the channels on the top. The squishy layer gives, and it's braced against that firm layer so it doesn't collapse on itself, making isolated compartments with this cool pinch structure, which currently looks like this. So that is a cage that is applying force from the bottom and the top to close those channels on the top. The exact same formula for quantifying as the standard method. All you need to know is the volume of your partitions. So how much volume is in each one of these straightaways, which I designed, the number of partitions, how many of them there are, and then you just count which ones glow blue, which ones don't glow blue. Throw in the formula, you got your bacteria. All good. Now, what I want to show you what this is going to look like. We haven't made it past the food coloring phase with this device yet. That's going to be next week, the bacteria. But I want to show you what the results are going to look like. So focus your eyes here, because remember, some are going to glow blue and some are not going to glow blue. And I'm going to turn off the lights really quick. There. So we've got, on this side, you can read it like a barcode if we stagger the channels. From this, you can read it like a grid from the top. Remember, we already know the volume and we already know the number. So you just count the blue, stick in the formula, you're all good to go. So where am I now? What have I got left to do? A lot. We're currently testing this device. The target outcome for this device is to test that proof of concept that can we physically close channels and make isolated compartments. The last phase of this test will be don't look. This will be a destructive test at the end of it. So I will be the last final phase of it. I will be taking the device and spearing it with live bacteria, and that the device will be gone. It's going to be dead. So I needed to design a new one anyway, because that device design was just a lot. So we're going to, next phase is design, fabricate, test the new version of the device. And our target outcome with this new version is going to be, I don't know why there's all one. That's 1, 2, 3, 4. The outcome of this one is going to be, hopefully, that is the phase where it passes all of the testing, the validation, the analysis to get to actually quantify the bacteria. Then, simple enough, make the sample pre-treatment strategy, take that ocean water, concentrate it down so I can pass it through this device and quantify it. And then I can finally go outside again. I've been trapped. I've been trapped in a laboratory for a very long time. So like the laboratory, with all laboratory work, it takes an extensive, incredible amount of support to get any of this off the ground moving. I have the most incredible team of undergraduate researchers, Michael, June, Renata, Sylvia, and Paula, I'm throwing these design challenges at them. I'm like, hey, how do we pinch this up? What do we do? They're like running back and forth between the 3D printer and doing it. There was a group of high schoolers over the summer with me that also worked on this project. This work is I do mostly at the Keller Lab in Bren, but I'm also working at CNSI over in engineering and the UCSB makerspace for rapid prototyping. My amazing guidance from my PhD committee, Dr. Arturo Keller, Dr. Scott Giuseco, Dr. Emily Dresser. All this stuff could not go on without the support from our Sage Davis is involved with like basically everyone's PhD, but especially lab people's PhD. And then Brian from CNSI, Emily, my collaborator from engineering, and then a lot of support from the laboratory mates on the 2L lab wing. So that's Levy, Becca, and Yuki, and also funded by Coastal Fund. So that is. Wow.
What a presentation.
Yes.
We've got time for a question.
Oh, no. That's my advisor. Yes. Oh, no.
Two bad questions, I'd say.
Yeah, those are rough questions. Those are rough questions. So the group with salmonella did it in 5 hours. So I was like super excited when I read that. The preliminary results I'm looking at, I'm just going to be happy if it's shorter than 24 at this point. There is some concerning data I found. I'm digesting this a little more. That is smaller, faster, or does it have more to do with the resolution of the detector? So that was going to be my presentation today. But I was like, deal with that next week. But that's my favorite question. Okay, so actually, I originally came to grad school because I was like, I'm going to do it for wastewater. This exact same test is used for wastewater, drinking water, and ocean water, storm water, all the waters. It's the exact same test. This was the least complicated matrix. So wastewater is like, oh, got a lot of stuff in it. Drinking water doesn't have enough stuff in it. Ocean water, perfect. And stormwater. I was an undergraduate researcher for a stormwater lab, and I'm particularly staying away from the rain because I'm tired of staying up till midnight being like, is the rain coming? Is the rain coming? So yeah, I don't recommend stormwater research.
Awesome. Thank you so much.